Let us do Brahmanand.
Hello students, in the previous session we discussed the steps of recombinant DNA technology. Recombinant DNA technology means it refers to the joining together of DNA molecules of two DNA molecules of two different species. And there are various steps. It starts with the isolation of DNA from the cell, from their respective cell. Which DNA we isolate? Yes, passenger DNA and the vector DNA we isolate from their respective cells. How? First we digest the cell wall, then we digest then the procedure is like that to digest the other macromolecules present with it, with the DNA molecule. Then for purification, the ethanol is the DNA, the extracted DNA molecule treated with alcohol, ethanol. Okay. Then next step is cleaving. Cleaving the passenger DNA and vector DNA by using the restriction enzyme, same restriction enzyme, but it is done separately. How and why? Why it is done? Yes, it is done to obtain the complementary strand of the passenger DNA and the vector DNA. So easily they can be joined. Next step is separation of the DNA fragments by using the gel electrophoresis technique. In gel electrophoresis, that is the movement of the charged molecules of different molecular weight. Okay, and next what gel is used in that gel electrophoresis? Yes, agarose gel is used for the DNA and RNA, and dye is used, that is maybe methylene blue visible dye or invisible dye, that is ethidium bromide, which can produce the fluorescent orange color under the UV radiation. Next step is the transfer of these separated fragments onto the nitrocellulose membrane, where the DNA probes are present on the nitrocellulose membrane. That again blotting paper is used with it is the blotting method, nitrocellulose membrane, where the membrane contains the DNA probes to collect the complementary DNA fragments. Then the fragments are collected. Now the fragments can be attached to the digested vectors to get the rDNA, recombinant DNA, or that recombinant plasmid, also known as chimera. Then it is introduced into the host cell for cloning. Host cells may be the bacteria or yeast cell, or it may be introduced directly into the cell of the organism in which we need this gene for the expression of the specific characteristic. That may be a plant cell, animal cell, any eukaryotic cell, or into the prokaryotic cell, into the bacteria or yeast we used to introduce for cloning. We will discuss. Then how we can know that? We discuss that whether it is recombinant became recombinant or not. How we can segregate the recombinant from non-recombinant? We use the insertional inactivation method, where the DNA are inserted into the selective markers or into the gene of the E. coli that express gene expresses the enzyme beta galactosidase, which produces the blue coloration in the bacterial colony. So that can be also within into that gene also the foreign DNA segment can be inserted, by which we can get whether it is recombinant or not, because in recombinant the blue coloration will not be produced because the insertion is done into the beta galactosidase gene. Okay. Now the time of cloning. How we will obtain the number of copies of that desired gene? So in this session we are going to discuss the gene cloning. Gene cloning can be done in two methods, that is host cell based and another is polymerase chain reaction based cloning where host cell is not required. What are the two methods? Gene cloning can be of two types, host cell based cloning, okay, or PCR, polymerase chain reaction based, host cell based or polymerase chain reaction based cloning. The polymerase chain reaction based cloning, this technique is discovered by Kerry B. Mullis in the year 1983. In this method, the gene means cloning means what? The making the large number of copies, identical copies. Large number of identical copies are produced of that particular gene. Which gene? Of course, the desired gene or the beneficial gene. The number of production of number of identical copies of a desired or beneficial gene is called gene cloning. And as we discussed, these are of two types. Host cell based, they are introduced into the E. coli or the other bacteria or yeast cell. Bacteria or yeast cells are used in which the cloning can be done. Okay, and in this method, the thermocycler is used for the thermal cycling, that is the heating and cooling method, on the basis of heating and cooling method. In this technique, the thermocycler is used. Thermocycler is the device. The reaction is carried out within the thermocycler. Thermocycler having a thermal cycling. Thermocycler having a thermocycling program. Thermocycling program, that is heating and cooling program. Heating and cooling program. Thermocycling program. Okay, where into which the desired gene, the target gene along with the other substrates are introduced. So this thermocycler is used for this reaction. Thermocycler having a thermocycling program is used for this program, is used in PCR, polymerase chain reaction. And here what are introduced? The target gene along with the substrates, along with the required substrates, What are the required substrates? Substrates are put into this thermocycler. So what are these required substrates? Of course, these are the nucleotides, we need the multiple copy of the required target genes. So we need nucleotides, we need the DNA primers we need, and we need the DNA polymerase, the enzymes, the primers, the enzymes, and the nucleotides. These substrates are required. Okay, here what happens in the first cycle, at the end of the first cycle, what will happen? It gets doubled. In the end of the, at the end of the second cycle, what will happen? It will quadruple, number of gene will be quadrupled, and so on. Here no host cell is used. It is this reaction of making the multiple identical copy of the target gene, desired gene, occurs without help of any host cell. Only the device is used and the required substrates are taken along with the desired gene. So, what are the, in this technique, it follows the, this PCR reaction is taking place by three reactions in the three steps. The three steps of this reaction, of the polymerase reaction, are the denaturation, then the primer annealing, and the extension. Denaturation, that is, denaturation means the double strands are they are to be made into the single stranded. They are denatured. Double strands, they are made to the single strand. And primer annealing, the how the primer, the DNA primer is annealed, joined to the 3' end of the single stranded DNA. Then extension of the nucleotides. Okay. Denaturation is done at the target DNA is heated. DNA is heated at 94 to 96 degrees Celsius, so that the two strands are separated. Okay, two strands are separated. Here what happens? Primer annealing. The oligonucleotide, the primers, they are annealed, joined with the 3' end. In primer annealing, it is cooled down to 54 to 56 degrees Celsius, so that annealing occurs. Then extension. The polymerase DNA polymerase enzyme, it extends the nucleotide sequence, okay, from the primer onwards. So extension, it is done at the, again at the higher temperature, that is 72 degrees Celsius. At about 72 degrees Celsius, it is the extension occurs. Okay, first of all the substrates and the target DNA, all the constituents are put into the thermocycler. That is, thermocycler in a PCR tube, that is Eppendorf. It is called Eppendorf tube. Okay, note it down. Into the thermocycler means what? There is a tube called Eppendorf tube or PCR tube. In general, it is called PCR tube. And into this, all the constituents are put into that. And let us discuss the each step in detail with the diagram. Step 1, the denaturation. Denaturation is the heating of target DNA. It is the heating of target DNA. Target DNA is heated to get the single strands of the DNA. Here what happens? The target DNA is heated at 94 to 96 degree Celsius, so that to obtain the single strands, that two strands are separated, so that resulting, what it results? It results into separation of two strands. So what happens? Separation of two strands. That means the double-stranded DNA becomes the single-stranded DNA. Each single-stranded DNA, here what happens? Each single-stranded DNA, SS means single-stranded, okay? DS, double-stranded. Each single-stranded DNA act as template on which the complementary strand is formed, okay? And how it occurs? So suppose this is the like this. The bond is here, okay? This is 3 prime to 5 prime. This is 3 prime to 5 prime. Okay, this is double stranded. How it becomes? It on heating at what temperature? The 94 to 96 degree Celsius. Heated at 94 to 96 degree Celsius. Then it is the denaturation of the denaturation. This step is denaturation. So what is obtained? It gives us the single strands. Here are the 3 prime to 5 prime, 3 prime to 5 prime. Next step is the step 1, we got the single strands. The step 2. What is step 2? Step 2 is the primer annealing. This is denaturation. Now step 2 is primer annealing. Annealing of the primer at the 3 prime end. The primer, the oligonucleotide, if the primer's length is of a few nucleotide. So oligonucleotide, 2 oligonucleotide DNA primers are annealed at the 3 prime end of each strand. 3 prime at 3 prime end of each strand. Why to the 3 prime end? Why not to a 5 prime end? Because the oligonucleotide primers are complementary of the nucleotide sequence of 3 prime end. Okay, that's why they are attached to the 3 prime end. Okay, like this they are attached to 3 prime end. So what are this is 5 prime to 3 prime, 5 prime to 3 prime. These are the primers. What are these? Primer are annealed. Here one primer, here one primer. Primers are annealed. 2 primers annealed at 3 prime end of each strand because these are complementary to the nucleotide sequence that are present at the 3 prime ends. Is that clear? Yes. So what I told, the primers are complementary to DNA templates. What are the DNA templates? These are the single stranded DNA strands are the templates. Primers are complementary to DNA templates. Template DNAs or DNA templates. And actually this mixture, after the denaturation, this mixture is cooled down to 54 to 56 degrees Celsius for 1 minute, for 1.5 minutes. This is 94 to 96 degrees Celsius. This is for 1 minute. 54 to 56 degrees Celsius while doing the in the step 2, 54 to 56 degrees Celsius for 1.5 minutes. Okay, and this is the step 2. This is step 1. Step 2 is, what is the step 2? Step 2 is annealing. Okay, and first of all, in the beginning of the step 2, the mixture is cooled down. as primarily it was heated, so cooled down to 54 to 56 degrees Celsius. Okay, now what is annealing? Anneal means join. Okay, here we can say these primers are annealed or hybridized. Annealed or hybridized. So what is written over here? Annealed. Anneal means join. But we can say anneal means join or hybridize. Okay, so they are the primers are annealed or hybridized. Next step, the next step is step 3. Step 3 is, next step is the final step. The polymerization is the final step. Extension or polymerization. It is extension or polymerization. In this third step, the extension or polymerization of the DNA strand occurs by here the Taq polymerase enzyme derived from a thermophilic bacterium called Thermus aquaticus. Taq polymerase enzyme derived from the Thermus aquaticus, derived from a thermophilic bacterium Thermus aquaticus in between two primers. What I am telling? The DNA strand, the DNA region is extended, polymerized by the help of Taq polymerase enzyme. We can say Taq polymerase enzyme which is derived from thermophilic bacterium Thermus aquaticus. Thermus aquaticus synthesizes the DNA region, synthesizes the complementary DNA strand. These are the template. So complementary DNA strands in between two primers, DNA primers, two primers. That means the two primers are extending towards each other. That means primers extending towards each other. In which direction? 5 prime to 3 prime. 5 prime to 3 prime. Other in 5 prime to 3 prime direction. So let us see what happens in the step 3. In the step 3, in the this was the 3 prime to 5 prime, 3 prime to 5 prime, and this extension process begins by heating the mixture at about 72 degrees Celsius for 1 to 1.5 minutes. Here the extension or polymerization in the third step, it begins by heating the mixture. Begins by the heating the mixture. Polymerization begins heating the mixture. Heating the mixture at 72 degrees Celsius. Okay, this is the third step begins. It may get confused. And I'm writing primer. Okay, write down this one is primer 1, this is primer 2. Okay, and heating at 72 degrees Celsius. Mixture is heated at 72 degrees Celsius. Okay, here what happens? The primers are extending. The primers are 5 prime to 3 prime. Here are the primers. Five prime to 3 prime. So extension, how it occurs? Extension, you see, this is the extension. Here 5 prime to, of course, 5 prime to 3 prime. Extension is done in the direction 5 prime to 5 prime to 3 prime. So this is how the extension of DNA region occurs from 5 prime to 3 prime. That means two RNA primers, the two primer DNA primers are extending towards each other. Okay, in the direction 5 prime to 3 prime. And this is the step 3, that polymerization or extension. This is step 3. Here I am writing step 3. Step 3 is the extension, or we can say extension or elongation and this is done at 72 degrees Celsius. This is for 1 to 1.5 minutes, 1 or 1.5 minute. Heated for, the mixture is heated for 1 to 1.5 minutes at 72 degrees Celsius. See, here you have to write down the polymerase chain reaction. These are the different, the first, second and third step of the polymerase chain reaction. Polymerase chain reaction or PCR. Okay, this is the in this manner the gene is the desired gene is cloned, and this thermophilic the Taq polymerase enzyme, the Taq polymerase enzyme is working in the presence of DNTP. The Taq polymerase enzyme synthesizes the DNA in the presence of DNTP, DNTPs and magnesium. What is DNTPs? DNTP is deoxynucleoside triphosphates and magnesium. In the presence of DNTPs and magnesium, the Taq polymerase enzyme extends or synthesizes the DNA region which is extended from the primers, the DNA primers. They extend from both the primers and they move towards extend towards each other in the direction 5' to 3'. And this is how the desired gene is cloned at the end of the first cycle then at the end of the second cycle the strands become quadruplet, means 4 DNA strands are produced, and so on. At the end of 30 cycle around 1 billion of the DNA strands are produced. Okay, how many? The gene can be cloned up to 1 billion in the 30 cycle. So this is called amplification. This cloning is also known as amplification. So at the end of 30 cycle, you may get about 1 billion copies. 1 billion copies of the gene. As we discovered, who discovered? The Kerry B. Mullis. Kerry B. Mullis discovered in the year 1983 and he received the Nobel Prize in 1993. He also received the Nobel Prize for the PCR in the 1993. And the PCR, polymerase chain reaction, occurs in 3 steps. That is, first denaturation, the double strands becomes single strand by heating the mixture. Okay, then the annealing means the primers are annealed with the DNA single stranded DNA strands at the 3' end, because at 3' end the complementary nucleotide sequence is there, means the primers are complementary to the nucleotide sequence present at the 3' end. Then in the extension or polymerization is occur in the 3rd step, where the Taq polymerase enzyme, which is derived from Thermus aquaticus, that in the presence of DNTPs, deoxynucleoside triphosphates and magnesium. In the presence of DNTPs and magnesium, the Taq polymerase enzyme that synthesizes or extends the DNA strands. Okay.